I'm going to cover the mechanism of crossing over during meiosis. So in a previous video, I alluded to the name of this mechanism. So it's going to be double-strand break repair. And I'm going to put by homologous recombination in parentheses. So let me start by diagramming a tetrad here. And I'm going to color code these. And I have some of these diagrams already drawn out for you, but I'm going to start nice and slow here because it can get pretty confusing. So here's a pair of homologous chromosomes. And when they align during meiosis, they can form a tetrad. Or when they're aligned during meiosis or paired up, we call that a tetrad. And here I'm showing a chiasma, which is the point of crossing over, an overlap of the position where two sister chromatids of a pair of homologous chromosomes overlap, a chiasma. And a result of chiasma formation can be a crossover. Now, it doesn't always end in a crossover, and I'll talk more about that in a moment. But we can end up with something like this. And the crossover is the physical exchange of DNA between chromosomes. So here we have this red from this chromosome over here was switched for this blue part over here. They were just switched. Okay, we see that there. So here we have a crossover. Okay, now I'm going to use colors, and let's assume that, okay, so the chiasma forms here, but in order for this process to start, we need a double-strand break to occur in one of these chromosomes. So let's say it occurs somewhere in there. In this sister chromatid of this chromosome, there is a double-strand break. Now, we're going to need to zoom in here and look at the DNA strands of these chromosomes. So, and I'm going to diagram that in a very, I guess, conventional way. By convention, we diagram when we're explaining how this process works. A certain type of diagram is used. Okay, so now I will so here is a DNA molecule inside here. So a segment of DNA inside that sister chromatid. And I'm going to put the hydrogen bonds. These little dashed lines represent the hydrogen bonds between the two strands of DNA. And keep in mind that, you know, this goes on for a long time, right? This chromosome can be hundreds of millions of bases long. And we're just looking at a small segment of it. And I'm showing you the 5' and 3' ends here, or orientations of these two strands. So now I'm also going to diagram the analogous region in this chromosome over here. I'm going to diagram that in red. Now, by convention, the orientation of the DNA strands relative to each other are diagrammed like this, with a 3' to 5' here, and this diagram here is going to have 3' to 5'. And the reason we do that is it's easiest to show how this double-strand break repair works when the strands are oriented like so. Okay, and last time I put the hydrogen bonds in here. I think it's the last time. Okay, so we have a segment of DNA in this chromosome over here and a segment over here. Now, in order for crossovers to occur between homologous chromosomes during meiosis, we need double-strand breaks to occur. So let's focus in on this one right here. Let's assume the double-strand break occurs here. And how does that occur? Well, during meiosis, double-strand breaks are normally really bad, but during meiosis, the double-strand breaks are programmed. And they happen randomly, or it's thought that at least semi-randomly, and they're caused by an enzyme called SPO11. So during meiosis, the very beginning of meiosis, there's an enzyme called SPO11. Okay, now SPO11 is going to be expressed. The SPO11 protein is expressed early in meiosis, and SPO11 is an endonuclease. So an endonuclease, now that's an enzyme that can, digest DNA or break phosphodiester bonds in the middle of a DNA molecule. Exonucleases, those enzymes can break phosphodiester bonds from the ends of a DNA molecule. Endonucleases can do so anywhere in the DNA molecule. Now, SPO11 is an endonuclease, and what it will do is it will find locations throughout the genome and induce double-strand breaks. And let's assume that it occurs here at this point in this blue chromosome. Now, the chromosome receiving the double-strand break is going to be called the recipient. And the one that is not getting the double-strand break will be called the donor. And the reason is because once this double-strand break is put in here, it's going to need to be repaired, and it's going to use information, it's going to receive genetic information from this one down here. The red one here, this is going to donate the genetic information. This is going to receive the information. So let's see what that double-strand break process results in. So I'm just going to diagram this blue chromosome right here. SPO11 is going to come in, it's going to make the double-strand break. Whoop, broke my pencil. Now, I'm going to put these little lines here. These little lines on the side are meant to show you that this DNA molecule goes very far in this direction and all the way to the end of the chromosome. In this direction, same thing, all the way to the end of the chromosome. Now, here's the double-strand break broken on both sides. So we have these loose ends of DNA here. I'm going to put a 3' here, 5' here. Again, these numbers are so important when talking about DNA molecules. Now, okay, SPO11 put the double-strand break into this molecule. And if you look at the notes, there's a little bit of more information about what happens next. So there's some advanced details about how some endonucleases, other than SPO11, and exonucleases process the ends. But the important thing to know is that the next step after SPO11 causes the double-strand break is the step called 5' end resection. And I don't know who came up with this word, but it's just a fancy way of saying that these 5' ends get chewed away. So we're left with 3' overhangs. So relatively long 3' single-stranded overhangs. So this 5' end right here is going to be lost, trimmed away, resected. I don't like that word resected, but that's what it's called. And same thing for this one over here. There's the 3' end, that stays. And then this 5' end, oh, it's nice and short. Okay, so now we have these single-stranded 3' ends, 3' single-stranded ends. So one here and one there. Okay, so now the next step is, here, let me diagram that again up here. This is the recipient chromosome with the 5' resected ends. and I will diagram it juxtaposed, I think that's a word, juxtaposed next to the donor chromosome, a DNA, the analogous segment of DNA from the donor chromosome. So after the 5' ends have been resected, what happens is the first, or the primary single end SE invasion, SEI, single end invasion step. And we're going to talk about a few of the proteins involved in this invasion step, I think in a later lecture. So, but what does this look like? So essentially, we're going to say this is the end that invades first. It doesn't matter which one you choose. I like to choose this one. And so what it's going to do is it's going to invade the donor chromosome, and it's going to look for homology. Or it's going to be used by other proteins to search for a complementary sequence in the donor molecule. And it's traditionally diagrammed like this, the search process. Okay, so now we have this little dropping down of the strand right here. I have some hydrogen bonds forming here. This is going to be the invasion step. And we see, okay, the strand's found some complementarity there. And we have, oh, do we have our orientations right? Let's see. Oh, nope, I have these wrong, right? This is 5 here. 5. Okay, so 5, 3 here, and then this is a 3' end here. It invaded 5', 3' there. Okay, this figure, you're probably a little confused now, but this is going to come together, trust me. Now this thing right here, the process of invading the donor template or the donor chromosome causes a displacement loop to form, a D loop. So D for displacement, displacement loop. This piece right here, this strand has been displaced during the homology search process or the complementary base pairing search process. So I need to diagram this end up here that hasn't really done anything yet, right? Okay, and I'm putting these lines here to indicate these chromosomes. This is just a segment of these 2 chromosomes. And okay, we have complementarity has been found here. So once that happens, okay, what can happen next? Well, now we have the second single end invasion step, or secondary single end invasion step. So what's going to happen is this one is going to now search the displaced strand for complementarity. And the end result is going to look something like this. Five, 3, 3 here. Okay, 3, 5. Okay, now let me put this guy in here, the donor chromosome. Okay, so we have some base pairing occurring here, and okay, so the secondary single end invasion step has found complementary here, complementary bases here. Now, let me put some hydrogen bonds in here to hopefully make the diagram more clear. Now, at this point, we see our first holiday junction. And essentially a holiday junction is where you have this intersection of DNA molecules and right here, and it's called a holiday junction because we can follow each of these to a double-stranded segment of DNA. So we have 4 double-stranded segments of DNA connected together through this Holliday junction. And Holliday is spelled like this because it's after a scientist named Robin Holliday, who predicted their existence. So 1, 2, 3, 4. Okay, so we have a single Holliday junction formed here. Now, after the second strand invasion step in the formation of the first Holliday junction, what happens is DNA polymerases come in here, and actually I'm not sure of the names of the polymerases, enzymes that participate in this process. So, and yeah, I wanted a different color. Oh, here we go. Pencil to show the new DNA that is being synthesized. So remember, DNA polymerases need a 3' OH group, so we have to find the 3' ends. And these polymerases are going to use, you know, the donor chromosome as the template. So let's go here. So we're making new DNA there, making some here. Now this step is, as far as I know, it is very poorly defined. Like we really don't really know how this next step happens. But somehow this is going to come up here and be ligated to that 5' end right here. So the new DNA is going to be made, and eventually we're going to get ligation to this 5' end right here. And same thing's going to happen over here. We're going to keep making new DNA, and eventually we're going to get ligation to that strand right there. So after the new DNA synthesis, after new DNA has been synthesized using the donor as the template and the ends have been ligated, we get our second Holliday junction, right? Because here we have this intersection of DNA strands right here, and we see double stranded DNA. So this is our second Holliday junction right here. Okay, so we've gotten pretty far through this mechanism. Now, at this point, what we have, and this is where I'm going to switch over to some diagrams that have already been put together for you. So we have the double Holliday junction stage, and now these need to be resolved, and they can be resolved in a couple different ways. So essentially, there are pairs of cuts that can be made by endonucleases. So we can cut here and here, so that's one pair that can be cut. The strand, the phosphodiester backbone of this top strand up there and this bottom strand can be cut. Or this can be cut and this can be cut right here. Or we can cut here and here, the purple strand and the red strand. Or the red strand and the blue strand. So we have 4 different pairs. We have 1, 2, 3, and 4. Now, these cuts can occur only in a couple of different ways. So you have to cut a pair. So for instance, if we cut this one up here, this one has to be cut. If you cut this one has to be cut. This one has to be cut, and so on and so forth. This one has to be cut. And you have to cut one of either this one or this one on this side, and one of this one or this one on this side. You know what? The easiest way for me to do this is put these in red. I'm going to put these guys in red right here. I think this is like I have it diagrammed in the notes, or like the diagram in the notes. So these arrows are red, and these are blue. Okay, so I have this diagram right here. I'm gonna get back to how these are cut in a moment. So we have these 2 chromosomes, and we went with the recipient is in blue and the donor is in red. It didn't have to be that way. It could have been the other way around. We have the chiasma formation, where you have all this double Holliday junction forming in here. Now this can be resolved in one of 2 ways. One way, we end up with a crossover. And in the other way, no crossover. So depending on how these how the cuts are made in the double holiday junction at the holiday junctions. Depending on how the cuts are made, we will end up with a crossover or no crossover. So essentially, if both of the blue pairs are cut, let's say both blue pairs are cut, cuts are made here and here, we get no crossover. If one red and one blue pair, so say this pair here, one red pair and one blue pair, we get a crossover. So it doesn't matter if it's this one and this one here, or this one and this one here, both of those will lead to a crossover. Now we also have another one where both red pairs that also leads to no crossover. But that's a little advanced and it's a little difficult to see this one, so I'm going to leave this one out. You'll notice that in the notes I suggest just ignoring this one. If you can see why when we cut both blue pairs we get no crossover, and when we cut one red or one blue, no, one red and one blue, we get a crossover. If you can see those two scenarios and understand that, then that's really great. That's really great. Then we can move on to other mechanisms. So let's first take a look at the no crossover situation. So the no crossover situation occurs when we cut these at these positions, cut the holiday junctions at these positions here. So I already have this diagrammed. So here we go. This is where the blue arrows were. The endonucleases came in and they cut at these locations. Now that leaves 5' ends and 3' ends on the DNA ends here, right? 5' and 3'. The 5' I didn't want to diagram the phosphate groups, but the 5' ends have phosphate groups and the 3' ends have hydroxyl groups. So those are prime sites or substrates for ligase. So we can see a 5' and 3' here, 5' and 3' here. This is the result of cutting those strands. So we've got a phosphate here, a hydroxyl here, phosphate here, hydroxyl here. Now remember, these used to be joined. So the phosphodiester backbone was cut, leaving a phosphate here and a hydroxyl here. Same thing for this one. Phosphate here, hydroxyl here. Now the same thing happened over here. Now ligase comes in, or a ligase, I'm not sure which one, comes in, and what it does is it joins this phosphate and this hydroxyl and seals, links these strands together with a phosphodiester bond. Does the same thing down here. So instead of this being linked up here, it's now linked to there. Instead of this one being linked down here, it's now linked to there. So we end up with something like this. So this right here now looks like this. See this purple and this red here? We have it linked together down here. And this red and this purple, it's now linked together right here. Same thing happens over here. So this red is now linked to this blue. And that blue is now linked to that red. Okay, now we can see why this doesn't lead to a crossover, right? Because look at the ends right here. This is blue, so this leads to the rest of the blue chromosome. We can follow this all the way down here, and this still leads to the rest of the blue chromosome. So there's been no crossover. Same thing down here. There's red in here. We follow this all the way over here. There's red over here. So the red chromosome is over here. The rest of it is to the left, and the rest of the other side of it is over here to the right. So there's been no crossover as a result of cutting the double holiday junctions, or the holiday junctions at these locations. This is also called resolving the holiday junctions. And that's one way to resolve the holiday junction. And this results in no crossover. We have some funny things going on in here, and we'll talk more about this in later lectures. But for the most part, the end result is no crossover. Now let's take a look at the crossover situation. So in the crossover situation, the one I'm going to go over is, let's say we cut these two strands of the endonucleases, cut here and here, and here and here. Now we could also cut here and here and here, here, but we're just going to look at this possibility first. So here and here, and here and here. I said first, but we'll only look at this one. If you can understand this one, then you should be able to figure out the other one. It should be obvious. Okay, so like I said, for the situation that results in a crossover, we cut where the red arrows were, here and here. That leaves a 3' end and a 5' end here, 3' and 5' here. And then we cut where the blue arrows were over here, leading to 5 and 3' ends here. And remember, 5s represent phosphate groups, 3s represent hydroxyls. And this one's easy. This is just like the previous scenario where this 5 is joined to that 3, this 5 is joined to that 3 by ligase. So we end up with something like this, where this red is joined to blue, and this blue is joined to red. However, here, this is something new, right? So this 3 used to be linked to this 5. It was cut here, so now this 3 is going to be ligated to this 5 down here. As a result, we're going to join this blue to this red. And it's going to kind of cause the diagram to need to twist a little bit. Right, see, we have this blue connected to that red down here. And I have this DNA molecule twisting a little bit, just twisting 180 degrees to show how now this strand right here is now connected down here. Same thing for this one. This 5 used to be here. Now it's going to be connected, it's going to get ligated down to that 3. So here we go. That 5 is blue. It's going to be connected to that red 3. So we have the blue coming down to this red down here. So in my diagram here, I hope you can see it. I've got this twist going on, but essentially this DNA strand is going back behind this DNA strand, which is in front. And we have a little twist. We have about 180 degree twists in each strand. Another thing to keep in mind, I forgot to mention this, is that I have these diagrams like, what's missing from these diagrams? The fact that these are double helices, right? So these strands are wrapped around each other, which is why it's so hard to resolve the double holiday junctions. They need to be cut and ligated. They can't just be pulled apart because these are double helices. The strands are wrapped around each other and are twisted around each other, and I obviously can't diagram that easily. So after ligase comes in and seals up those broken phosphodiester bonds in this particular pattern, we can see that a crossover has occurred, right? So here's the remaining part of the blue chromosome over here to the left. We follow that. It's now linked to the red chromosome over here. Same thing for this one. So we have red chromosome going that way. It's now linked to the blue chromosome going that way. Okay, well, I hope you can see how cutting here and ligating them like this results in a crossover. But cutting at both blue arrows here results in no crossover. I hope that wasn't too confusing, but that's essentially how, well, what is it? It is how crossovers occur during meiosis, and now we know that sometimes this chiasma and the double-strand break repair model by homologous recombination sometimes leads to a crossover and sometimes leads to no crossover. Okay, so that's it for lecture 17. There's a lot of stuff in there. Please check out the typed up notes and give the problems a try, and let me know if you have any questions. Okay, thanks.